today we're going to start talking about some of the specific details of the innate immune response. And so I'm kind of thinking about, in general, aspects of this part of the course as where we're kind of building the immune system a bit. So we're going to see and build aspects of the innate immune response today as well as next week. I will also tell you that some students, by the end of this class, because of some of the stuff that comes up in this particular class, start freaking out about like exams and stuff, and they're like, oh my gosh, how could I ever handle that on an exam? It's okay. Totally okay. But that's one reason why this weekend I'm going to make sure all my old exams, and in fact even old problems on this exact topic, are posted. And the inquisitive also is about this particular topic. So if there are a couple places where you feel like, oh my gosh, the details today, know that you can actually both have the inquisitive as on purpose to help you with those details, and B, you can even look at sort of ways I have addressed it on old exams once I have those posted by the end of the weekend. So just be ready for that. Okay. So I mentioned to you last time the fact that we can divide up the immune system into lots of different categories, particularly thinking about these different layers of immunity, either the barrier defenses, the innate immune response, or the adaptive immune response. And for today and for next week, we're really going to be zooming in on innate immunity. We can do other types of divisions to kind of make sense or classify different aspects of the immune system as well. And the aspect of the innate immune system that we're going to talk about today, here you can see I listed as the humoral response. That was originally a name that was given when it seemed like the liquid of the blood was active. And really what it means is that we're looking at some proteins. And so today we're largely thinking about proteins of the innate immune system that are basically pre-made. Your body has them right now. And so if a microbe were to enter your body, those innate immune proteins could start to act immediately. So this is sort of the first thing that might happen if a microbe were to enter your body. You don't actually even need to do any transcription or translation. These proteins are already ready to go, waiting in your body to start trying to combat microbes. And so you can see that this is really part of this very immediate response, where perhaps our pathogen will invade. And we have these preformed proteins that might be able to eliminate the pathogen, and it might be done there. We might not ever get to any further parts of immune responses. We might not ever get to really much of any tissue damage, much of any disease. This is going to really be helpful in acting against those microbes. Even when the microbe is not totally controlled by these proteins, which is the part of this figure that I whited out, and I'm not showing you here because we're doing that later. These proteins are really important in terms of knocking down numbers of microbes and making sure that those microbes are not increasing exponentially so that we have a chance. Generally, these innate immune proteins are going to be acting against extracellular pathogens. They are going to be acting against things that are outside of cells, either because they live outside of cells or because they have not yet made it to inside of cells to be intracellular pathogens. And so the things that we're looking at right now are largely, like I said, early and preformed, as well as largely acting against extracellular pathogens. This also is a place where my nice, perfect little description of layers of the immune response already starts to get a little weird. Because in fact, one thing to know about some of these these innate immune proteins, sometimes also mentioned as antimicrobial peptides, is that they are a common feature at the barrier organs. So one of the things that's great about the barrier organs, like skin, gut, lungs, etc., is that they all have a lot of antimicrobial peptides. So that we can immediately start to combat microbes at those barriers because those barriers are in contact with everyday life. They're not only found in barrier organs. There are many, for example, that circulate in the blood. But this is already one place where sometimes the distinction can get a little gray. You'll see here that I have these listed as antimicrobial peptides. And in reality, there are tons of different antimicrobial peptides. I'm going to highlight some particularly interesting or important antimicrobial peptides, but know that the ones I am telling you about are examples out of a really big set of potential proteins or peptides. One that I don't really have a specific slide about, but that I will point out here just because it's cool and I like it, is one called Lactoferrin. You can see that Lactoferrin is in a number of barrier tissues, though it can be in a number of other places as well. Lactoferrin works by just binding all your iron. Microbes need iron to reproduce. They want to steal it from you. So basically, lactoferrin just hides all the iron so the microbes can't have it. It's really fun, actually. When microbes try to steal your iron, technically that strategy is called iron piracy. And so they're trying to, so you just have a protein that basically soaks up all your iron so the microbes can't get it. There are tons of different ways that that can work. You might imagine that lactoferrin is probably going to be pretty broad in its action against many microbes. Other antimicrobial peptides can be a bit more specific. I always feel weird when I show this slide because this is actually a great experiment. It makes a good, important point, but I also worry that you're going to somehow tell Dr. Miller that I told you to do this. I did not, do not do this. No, 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 no. In this experiment, we have a person who actually put some bacteria on their bare hands, on their skin, either Staph aureus or E. coli. Staph aureus being a gram-positive organism, E. coli being a gram-negative. And then the individual let their hands just sit for 30 minutes. Then those fingers went onto a culture plate. And the idea was to see if that Staph aureus or that E. coli remained on the skin. When they looked, they saw that Staph aureus was still grew, while the E. coli did not. And the idea here, and in fact the reasoning behind this, is that you have a large number of antimicrobial peptides on the skin. And in this case, these antimicrobial peptides tend to be more specific. They're not necessarily acting against all microbes. They don't act against Staph aureus and many gram-positives, but they might act against gram-negatives. So depending on which of this huge list of antimicrobial peptides we are talking about, they could be very broad, like lactoferrin, or they could be more specific, like you might imagine in that example I have just shown you. When immunologists think about antimicrobial peptides, there are a few really critical antimicrobial peptides that we think about. One of the most famous examples of an antimicrobial peptide is called lysozyme. Note that lysozyme has zyme at the end of its name. That's because it's an enzyme. It has some enzymatic activity. Later in the semester, we will talk about an organelle called the lysosome. Lysosome, S-O-M, and lysozyme, Z-Y-M, are not the same thing. One's an organelle, one's an enzyme. So be careful on that. And lysozyme specific enzymatic activity is that it cleaves a particular chemical linkage called a beta-1,4 linkage. The reason why you care about that is that different bacteria will have specific cell wall structures, as shown here, that can include this molecule peptidoglycan as a major part of the cell wall. Different microbes have different amounts of peptidoglycan, but many of them do have peptidoglycan. Lysozyme's specific enzymatic activity breaks a particular chemical linkage, the beta-1,4 linkage, in peptidoglycan in order to destroy the prokaryotic cell wall. And once that cell wall is destroyed, that can make it very easy for the microbe to be lysed by osmotic lysis. And so this will lead to lysis of the cell. Lysozyme is found in many mucosal secretions. It's in many of your different barrier organs. Two of the best places, if you decided what you really wanted to do was purify a whole bunch of lysozyme, the two places that you would actually be able to get it from the best are saliva and tears. So there's tons of this trying to sort of inhibit potential microbial growth in saliva and tears. Another really critical group of antimicrobial peptides are a group of peptides known as the defensins. Again, there are many defensins. I'm just going to say defensins. But there are, for example, people whose lives' work is human beta defensin 3 or something like that. And I'm just going to say defensins. You can see that they can be made by a number of different cell types, some immune cells, like our neutrophils, some sort of more non-immune cells. You can see that they can be present in many different tissue types. Many of them are barrier organs, though we can see them in some other places as well. And what you can notice is that in most situations, they are made constitutively, meaning they're being made all the time, 24/7. So you always have them to potentially stop microbial replication no matter what. This is sort of that, first few minutes after exposure. These are going to be helping you against microbial replication. That's why they're constitutive. They're there all the time, ready to go. One of the places that they are very commonly found or very famously found is in the GI tract. So if you look at a GI tract, you have these villi as part of the intestine, and there are these sort of deeper areas called crypts. Many of the cells in the crypts actually make tons of antimicrobial peptides, particularly defensins, in order to stop or control microbial replication in the GI tract. Defensins have a mechanism of action that's also pretty straightforward. So defensins really just act because they are little charged proteins. So you can see they are these proteins with a positive charge. And the way that they work is that they are able to actually insert into plasma membranes and make pores. So they poke holes in plasma membranes. to allow for osmotic lysis. Basically, water comes in, cytoplasm goes out, the cell no longer has structural integrity, the cell's dead. It's really all that happens with defensins. Sometimes people look at defensins and say, okay, I get this. I get how that kills a cell. I can see how this would be useful against microbes. But what I don't understand is how in the world does this not kill all of my other cells too? How can my intestine make some defensins, kill the microbial cells, and not kill the intestine cells? Because my cells have plasma membranes too. And the way that that works actually is that our cells do a lot of regulation of the phospholipids that are in the plasma membrane and move all of the phospholipids with negative charges to the inside of the plasma membrane. When you were in Bio 250 with Dr. Dunaway, you might have learned about flipases. That's what flipases are doing, is that they're making sure that all of the negatively charged phospholipids are on the inside. We use that for lots of reasons that I am not going to deal with right now, but it just so happens we do that. Microbes don't sort of organize their phospholipids in the same way. They've got negative charges on both the in or the outside. And so these positively charged defensins can bind to those negative head groups on prokaryotic cells and destroy their membranes while not destroying the membranes of our eukaryotic cells because of the sorting we have done on those phospholipids. I mention this because most of the time this is what we say about defensins, is we're like, oh yeah, they just insert into membranes and make holes and ta-da. And I know that I have had many times where students have been like, that seems pointless. Why would we do that? And so this is the reason why we would do that. One other thing to point out about antimicrobial peptides in general is that sometimes they can also act not just in terms of microbial replication, but they might also stop different types of microbial processes that are involved in disease. So many microbes induce disease partially because of specific proteases. So many microbes have proteases that can cleave stuff of yours and cause different kinds of disease. I will admit, usually that does help them replicate too, but we're going to think about it more in the disease sense right now. We also have antimicrobial peptides like alpha-2 macroglobulin, which is pretty cool. Alpha-2 macroglobulin basically has a protease substrate in the middle of it. It's like bait for proteases from microbes. And if a protease tries to cleave alpha-2 macroglobulin, the protein undergoes a conformational change and closes off the protease and inactivates it, but also basically it sequesters that protease. So you can see the protease tries to cleave alpha-2 macroglobulin, and alpha-2 macroglobulin actually sequesters and inactivates that protease. So now that protease cannot be involved in destroying other targets and cannot lead to disease. And so we've evolved a lot of different ways that we can target potentially problematic microbial processes, which in some cases could be a better strategy than trying to get rid of all microbes ever. Just get rid of anything that might be dangerous. And so this is another strategy we see with antimicrobial peptides. Sometimes students will see different mechanisms that we learned throughout this semester. And one of the places that they will sort of be a little confused is, okay, awesome, we have a semester's worth of all these fancy aspects of our immune system that are protecting us from microbial infection and protecting us from microbial disease. Why the heck do we ever get sick? Why do we need so many? These seem like great things. And so I just want to also point out that we are in an evolutionary arms race with microbes. They are trying to evolve to evade all of these things. There are plenty of microbes that evade antimicrobial peptides, either by destroying them or by exporting them or all sorts of potential mechanisms. And so I'm not always throughout the semester going to show you evasion mechanisms. But basically everything we talk about this semester, there are microbes that have evolved ways to evade. So I wanted to also point out, because some way when we finish some of the things we talk about today, students are like, why isn't that not it? Isn't this, we had some cells the other day, we have some proteins, that seems like enough. And part of the reason why this is not enough is because of microbes trying to evade everything that we're doing. If I showed this slide to an immunologist, one of my colleagues from grad school, their mind would go to a very specific place. They would be like, oh, I know what she's going to talk about today. Because when we think about proteins of the innate immune system that help to combat microbes, there is one particular set of proteins outside of the ones I've already told you about that are super famous. And so everybody, any immunologist who saw this would be like, okay, that's what this is about today. And the rest of the time today, I'm going to talk about that group of proteins. These proteins are known as the complement proteins. There is much to be said about complement proteins. So when I started taking immunology, when I started TAing immunology, complement was really like the short straw that no one ever wanted to think about and talk about. And we would all be like, oh, really? Do we even have to talk about it? Like, we would joke as TAs, like, who gets the short straw of having to actually talk about complement? No one wants to do this. Everyone hates it. It was described really early in the history of immunology, and sort of seemed really, I guess, old and dusty when I was starting grad school. What we have realized over the years, in a lot of ways, this is a little perspective piece talking kind of about this, are all of the things that complement is actually really important in the body that we maybe never thought of before. So this is an example of actually an eyeball of a person with macular degeneration. All of these little clumps are actually the things occluding the eye to stop this person's vision. They are all clumps of complement. We have realized that complement is actually a major contributor to pathology in arthritis, particularly osteoarthritis. There have been some really beautiful papers showing that complement is actually critically responsible for synapse pruning in the nervous system, and on and on and on. And so complement keeps getting related to things that maybe we didn't even originally know were immune related. And its importance is particularly sort of obvious. But you will notice there are a few kind of really weird old-timey phrases that come up when we talk through complement because it was described such a long time ago. I'll also tell you why it has the name complement. Once upon a time, people, and you'll see why this is, people realized that if you were trying to do certain experiments with antibodies, you could add this stuff and it would make the antibodies better. It would complement them, would improve them. And so this was known as complement, or these proteins that sort of improved or helped out antibodies, which is where the name came from. We often talk about, we'll sometimes refer to complement as C prime. So if in a paper you ever see something that's just like, and then we added C prime, that's immunologists speak for complement. And we often talk about this thing called the fixation of complement. I don't know why they picked that term. That term was picked I don't know, probably before my grandparents were born. Definitely before my parents were born, I would say. So I don't know why they picked it. But the basic idea for complement is that there is some complement protein. Most of the complement proteins have very creative names, like C3. And those proteins are cut by a protease. You can see that protease activity happening here, to make a couple of different fragments. And one of those fragments will be small and will float away and do something. And one of those fragments will actually get attached to the microbe. And often when I think about something fixing complement, that means a bacteria has complement fixed to it if we've attached a piece of a complement fragment to the bacteria. And so really the very broad overview of what we're going to see when we think about complement is a bunch of proteins getting activated by a protease, part floating away and doing stuff, part getting stuck on things. Sometimes people can get a little overwhelmed thinking about what's going on with complement. I really like this figure from your textbook because it helps us understand big picture what's going on with complement. So big picture what's going on with complement is that there are 3 different ways it can get started. There are 3 different initiation pathways. All 3 of those initiation pathways then meet up at one common similar point. So, and that point, as described here, is activating something called C3b. So specifically cleaving C3 into C3a and C3b. So, but we'll just say getting to doing something to C3. So really, the initiation pathways are how do you get to C3? Then, once we've gotten to C3, there are a few different ways we can deal with the pathogen. So once we get to C3, we have a few different ways we can deal with the pathogen. So when you're thinking about different parts of the complement cascade, one of the things you should be aware of is, are we talking about initiation? Are we trying to figure out which of these top things we're doing to get us to C3? Or are we thinking about an effector function? It wasn't until I was interviewing for professor jobs that I learned that people who are not immunologists don't use that word. We use it so much that I'm like, what do you mean? Everyone knows that word. Effector function means how you do stuff, how you have an effect, how you get rid of the microbe. So one thing to be aware of as we talk about different parts of this is, are we talking about an initiation type of part, or are we talking about an effector function type of part? Each of the different effector functions that we see could happen following any different initiation pathway. So you could go like here and then here, you go here and then here. Usually you go here and then like multiple of them, okay? So the way that I'm going to talk through this today is I'm going to first talk through one initiation pathway and get us to C3. Then we're going to talk about the effector functions, sort of the things that could happen. Once we've done that, I'm going to go back up to the top and show you the 2 other ways we could have gotten to C3. So that's kind of the overall view of what we're doing here. This is a nice figure from your textbook that gives you an overview of exactly what I'm going to be talking about, this exact process, and it divides it up in the same way. Just for your studying purposes later, I've put in the same concept figure from a different textbook. It has some pictures instead of just words. So when you're studying later, whichever of these two figures makes more sense to you, they're both here for you. So the version of complement initiation that I'm going to start with is called the classical pathway. The classical pathway was named the classical pathway because it was the first one we discovered and understood. So it was kind of the first way we understood it. We now know that this one was actually probably the last one to evolve. And in a lot of ways, it's probably not the one that happens first. It's probably not the one that even happens last in your body. But it's the one that was described first. To me, it makes the most sense to start with description of the classical pathway. The classical pathway is unique because the classical pathway starts with an antibody binding to the surface of a pathogen. And the antibodies are drawn here as these Y-shaped proteins. And we are going to see so much more about antibodies going forward. So you can see these Y-shaped antibody proteins. A thing that is really, really, really important to know is that antibodies are products of the adaptive immune system. They're not usually thought of as part of the innate immune system. So the classical pathway actually requires the adaptive immune system to do something, even though we are totally in an innate immunity day. So one of the things that's sort of unique about the classical pathway is that it starts with an antibody binding to our microbe. And so you can see that antibody binding here. The specific geometry of the antibody binding to the microbe is actually kind of important. But we'll get there. Then the complement pathway is initiated with the first of these complement proteins. And the first complement protein that's involved in the classical pathway is called C1. See, because of the way the numbers go in this one, that's why I like to do this one first. We start with C1. So there is this protein that's called C1. And you can see a cartoon version of C1 from your textbook on the left, as well as a microscopy image of C1 on the right to show you that that is really what it looks like structurally. And we usually describe C1 as looking something like an upside-down bouquet of flowers. So you can see that looking at C1. C1 can bind to that antibody that's on the surface of our pathogen. So we started the classical pathway with an antigen, or the antibody bound. C1 can now go ahead and bind to that antibody. Sort of the geometry, again, is a little important just to make it fit C1, but we're not going to get super into that. When C1 is bound to antibodies, as is shown on this slide, it gains the ability to act as a protease. And when it gains the ability to act as a protease, it cleaves the next protein in the the classical pathway. The classical pathway is where you learn that immunologists can't count. So we'd love to go from C1 to C2, but we don't. Instead, C1 activates C4, C4 activates C2, and that activates C3. So it's actually 1, 2, 3, 4. It just 4's in the wrong place. So you can see this on these slides as well. So the first thing that will happen is C1 will bind. That will allow it to interact with C4 and C2. It's actually 4 first, then 2. Little pieces of them. You can see that C2 was shown here before it was cleaved. It's a little smaller after it's cleaved. Same thing with C4. C4 and C2 will now both have pieces that get stuck or fixed onto the surface of the microbe. When these are stuck to the surface of the microbe, they form a protease. And that protease cleaves the next one in the line, C3. So you can see that here. Once we have C4 and C2 on the surface of the cell, we can activate C3. Oh my gosh, C3 was the middle part I already told you about before. We're done. We're there. We got there. All good. It does turn out that there is one other way that you could start the classical pathway. And that's a good thing, because the classical pathway, as shown here, requires an antibody to start that process. And antibodies are part of the adaptive immune response. Maybe you don't have a great antibody response at the beginning. The first time you've ever seen a new microbe, you may not have an antibody response. Fortunately, there are some other proteins. A famous one is called C-Reactive Protein that can bind to the surface of the microbes and then bind to C1 and do the same thing. So C-Reactive Protein is made by the liver, as are most of our complement proteins. It can bind specifically to microbial surfaces and start this process as well. Yes? You know, it's not less frequent, but it was kind of described later. We didn't really understand it as well, and it's sort of not the famous way of doing it. So C-reactive protein, like I mentioned, and many of these other complement proteins are made by the liver. If you are going to have blood work done, if you remember, we talked yesterday that you might have someone check your white blood cell counts to see if you have too many white blood cells. Sometimes you also will have your C-reactive protein levels checked, again, to see if you are making, if your liver has been turned on to start some kind of immune response. So that's another reason to mention C-reactive protein is often seen in blood work. Yep. No, it can occur anytime. That C-reactive protein does have the ability only to bind to microbial surfaces. So once we have C3 activated, we can now do something about our microbe. Your textbook shows 3 different potential effector functions that can happen, and I will talk about all 3 of those. I am going to briefly mention a 4th, just to say that it's a thing. I'm not going to go into further details on it. So the one that many people think a lot about, when I think of complement, I will admit this is usually the first one my brain goes to, is formation of the membrane attack complex, or the MAC. There was some song when I was younger about return of the MAC, and now it'll be in my head for the rest of the day, since I had to return to the MAC slide today. So once C3 is active, it can activate C5, which is not shown here. When I was actually putting these slides together, I was unimpressed with how they dealt with C5 in this version of your textbook. They just kind of were like, and didn't really get into it. Usually C3 activates C5. I actually sometimes draw this layer as the C3, C5 layer. Whatever. C5. It's a thing. It, just like every other piece of one of these complement proteins, gets cleaved into two pieces. So it will get cleaved into a small piece and a big piece, an A and a B. You can see that here as well for C3. So C3 will go into C3A and C3B. And C3B is actually the one that was bound to the surface of the microbe. C3A is the little one that floated away. The exact same thing happens with C5. C5 gets stuck to the microbe. C3A or C5B gets stuck. C5A goes away. So here you can see C5B is getting stuck to the microbe, and it brings along its friends 5, 6, 7, 8, 9. Again, we're basically counting for this one. That's why I like to do this one first. And that makes a pore in the surface of the microbial cell. So C9 is this little pore-forming protein. We get lots of them and make a pore, which are actually easily visible under a microscope. So here you can actually see pores in a bacterial cell that have been poked by C5. You can also see a cylinder right here on the side of this microbial cell, this little tube. That is C5 in a side view on this cell. And as a result, our bacteria is lysed. So we have lysis of our cell. So one thing that we can do with complement is we can basically put all of these little sticky notes on the bacteria and eventually start lysing at that sticky note spot. We can also do some other things. So if you recall, I mentioned that C3b was getting stuck to the surface of our microbe, as was C5b. And I mentioned, oh yeah, there are those little pieces that float away. C3a and C5a. C3a and C5a also have a particular role. One thing I forgot to say on this slide is that if you go look at that animation I mentioned, the chapter 2 animation that's on Moodle, or that's linked from Moodle, it basically is starting from here and going through here. And it's just a video of each of these steps with someone telling you about them. So you can see it again there. But C3a and C5a, the little pieces that are cut off, have an important job. And those two proteins can induce inflammation. So they can do things like change blood vessel permeability and change blood vessel size and generally start an inflammatory response. And so we'll see changes to kind of vessels, largely to help recruit in more immune cells. We can sort of have these smaller proteins act as alarm bells that say, hey, we got a problem right here. Can you please send over some white blood cells to this location? That's basically what C3a and C5a are doing. They can actually act on a number of our different innate immune cells by binding to receptors, but the overall role of them is to lead to inflammation. We're going to talk a lot more about other aspects of inflammation next week. Sometimes people refer to C3a and C5a as the anaphylatoxins. So if you see that word, it just means they're little proteins that induce inflammation. It's a word that every so often you might see. So I'm just going to tell you what it means, even though I'm not going to be super into it. The third thing that complement proteins can do is called opsonization. Opsonization is one that is near and dear to my heart. Those of you who've taken micro have already heard my opsonization story, but you get to hear my opsonization story again. I have in my office I still have my first ever immunology exam from my first undergraduate immunology class. And on that exam, there is a place where there were some terms and we had to define them. And opsonization was one of the terms, and I got it wrong. And the reason why I got it wrong was because my professor had actually taught us a sort of analogy to help us remember opsonization. And I could remember the analogy but couldn't think how to describe it otherwise, so I just wrote the analogy or something related to it, and I got it wrong. So I'm going to actually tell you that same analogy now to help you understand opsonization, but don't write it on the exam. Lots of people now write me random jokes related to it, next to writing their real answer. The other reason why this actually cracks me up is because I've been telling that story for years when I teach immunology. And a former member of my lab went to graduate school in immunology at the same university where I was an undergrad. And in one of her graduate school courses, the person who taught it to me taught it to them with that same analogy. And she sort of like couldn't handle herself, that she was in that same class hearing that same analogy. It was crazy. So people still use this analogy to this day. So first I'm going to tell you what opsonization is, then I'm going to give you the analogy that we can kind of see how it all works. So with opsonization, we are thinking about those fragments of our complement proteins that are attached to the microbial surface, like C3b. So you can see here the proteins being attached to the surface. And whenever we think about opsonization, because it does come up in some other places in immunology, part of it means something getting coated with stuff. Whatever the coating thing is, in this case, it's complement proteins. Those coating things can be bound by a receptor. Here, it's a complement receptor that's on some kind of cell. So we've got our complement proteins binding to a complement receptor on a particular cell. Oftentimes it is a macrophage. And now that macrophage will phagocytose that bacteria and destroy it. Basically what has happened here is that we have marked this bacteria to be phagocytosed. We have marked it for phagocytosis. The way that my professor explained it to me was that we have put a coating on that bacteria to make it more likely to get eaten. Or we've put a coating on it to make it look extra tasty. And so opsonization is like butter. It's like putting butter on something to make it more likely to get eaten or to make it more tasty. And yes, I wrote butter was the definition of opsonization on that exam. So it's like butter in that it's a coating that makes something more likely to get eaten and labels something as a thing to be eaten, but it is not actually butter. The other thing that I will tell you that does happen following complement fixation also has to do with complement receptors, just like opsonization does. It's slightly different complement receptors, but the sort of thing I just want you to know is that there are also some complement receptors that are on adaptive immune cells, and complement can also start to activate the adaptive immune response. So we can also help the adaptive immune response by coating things with complement. So we will also turn on adaptive immunity, although since we haven't really talked about adaptive immunity, we'll say enhance. I'm not going to say any more than that. I just want to point out that yes, in fact, that can happen. So at this point, we have started with one initiation pathway for the complement cascade. We've gotten to our section of C3 where all of these complement initiation pathways come together. And we have gone through multiple different ways that we could eliminate the infecting pathogen. As your textbook says, and as I mentioned before, the classical pathway is likely the last to be activated. And we now understand that if we look in terms of evolution, it is probably the last one to have evolved. In fact, if we look at different groups of animals, only some can do the classical pathway. Only mammals, birds, reptiles, amphibians. We're only seeing classical pathway there, while these other pathways of complement initiation are found throughout animal phyla. So in fact, they probably evolved first, and classical pathway was like an add-on. I still find it easier to explain classical first. So one of these pathways, you can see there's a nice figure in your textbook, figure 3.35, that talks about this as well. I liked this one better, but there's a similar figure in your textbook. It's called the Manos binding lectin pathway. Sometimes also just referred to as the lectin pathway. I've asked this question of students many times in the past. The answer is almost always no. So if the answer is no, that is okay. But lectin is actually a general biochemistry term. Does anyone know what a lectin is? I'm not seeing anyone like jump at answering. No. So a lectin in general is a protein that binds a carbohydrate. We'll see a few different lectins this semester. Specifically, the lectin in this case is called Manos binding lectin. It binds to a special carbohydrate. Guess what that carbohydrate is? Manos! So this pathway actually starts with a protein called Manos binding lectin, or MBL. So the lectin pathway starts with the lectin, MBL, Manos binding lectin. Those manoses are found really frequently on the surface of microbial cells. The reason why I didn't show the picture of this from your textbook is for some reason your textbook didn't show you any manoses. It was just like, Manos binding lectin magic. And I'm like, no, there's manoses. So usually there are manoses on microbes. And so we've evolved a protein that binds to those manoses, the Manos binding lectin. You can remember the name. You pretty much got it down, what it does. If you look at Manos binding lectin, which is shown here in green, does it remind you of anything, or does it make you, start to think about anything? Yeah, Jameer.
It's like C1.
It looks an awful lot like that C1 protein that you saw with the classical pathway, right? Looks like an upside down bouquet of flowers, just like C1. Guess what? MBL, when it is bound to Manos, acts exactly like C1. So in fact, what happens with MBL is that MBL turns on C4 and C2, and everything else happens exactly the way we've already talked about it. There, you already learned the MBL pathway. It's a pretty straightforward one. Life gets slightly trickier with the third pathway. And the third pathway is known as the alternative pathway. The lectin pathway is pretty good. It, it works when any time a microbe has Manos on it, which is like most of the time. But, we also need to And so you don't have to have a pre-made antibody. No, we haven't seen any situation where we had to have a cell come and do something. We didn't need to wait for any transcription or translation. So these microbes are going to be able to be attacked fast with these pathways. What we've realized is that the alternative pathway is probably the first to have evolved, and the alternative pathway is really fast. The alternative pathway is happening in many cases within like a minute of that microbe coming into the body. I guess that's a weird arrow. That's a better arrow. But the trick about the alternative pathway is that the alternative pathway's initiation process is a little, strikes people as odd. This is going to be one of the first places where you learn about something this semester where you're going to be like, that is not intuitive, and that is weird, and I don't know if I totally believe you because I don't like that. So I know that some parts of the alternative pathway are a little bit uncomfortable. And to fully understand the alternative pathway, we have to talk the teensiest bit about the C3 protein and some aspects of its biochemistry. So this is what the C3 protein looks like at rest. The C3 protein has this bond inside of it called a thioester. So we have this thioester in the C3 protein. That thioester has a really weird angle in it. It is very strained. It's under a lot of stress when the protein is all folded the way that it's supposed to be. Sometimes, because that thioester is so strained and stressed, it just breaks. And so sometimes C3 spontaneously breaks. When C3 spontaneously breaks, we have this bond free, and that bond is a really good nucleophile. It wants to attack something. It wants to react. It's really, 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 really reactive. Sometimes it reacts with water, because water's the closest thing. And if it reacts with water, then you get this weird piece of C3 that isn't attached to anything, and basically no one ever cares about it ever again. But sometimes it doesn't react with water. Sometimes it might react with an R group with an OH or an R group with an NH2, so a hydroxyl or an amine. And in that case, you'll see the C3 bound to the hydroxyl or the amine. If you think about biological molecules that you know, do any of them have any hydroxyls or amines? Anybody? Can you think of anything that has an OH or an NH in it? Yeah. DNA. What were you going to say, Kat? Amino acids. Sugars. So basically, all proteins, carbohydrates, DNAs, every biological molecule. So if there's something other than water as the closest molecule, say, oh, I don't know, a cell. If this happens near a cell, this is going to automatically attach itself to the cell. And so right now, C3 is automatically breaking and attaching to cells in your body because of this weird biochemistry of this thioester. And you can see this happening here as well. C3 can spontaneously cleave and can be immediately attached to a microbial surface. So if there happens to be a microbe around, it's going to get C3 stuck to it almost immediately. And so C3, or the alternative pathway, starts with C3. activating itself. There are other proteins that help this process out and stabilize it. I'm going to show you the next 2 slides very quickly and just point out a couple of quick things. Don't stress about this. There are other proteins that help stabilize C3 in this part of the alternative pathway. These can include things like something called factor B or factor D. This can also include something called properidin. What you can notice is that these proteins have very different types of names than the ones that are involved in the other parts of the pathways. And so if you hear about factor B in complement, that just sounds so different than all of the other ones. That should hopefully trigger in your mind, that's alternative pathway. That's that weird one. Because it's the one with weirder names, not the official sort of C1, C2, C3 names. And these will all come together to help C3 B start to be stable on the surface and cleave more C3b. Sometimes you get fun names, and this is actually true. There is a protein as part of this that's called C3bbb, and all sorts of fun stuff like that. And one of the reasons why the alternative pathway is so important is that if we start the complement cascade through any one of these pathways, once we get to C3, we can actually amplify more with the alternative pathway. So maybe there was only one antibody that bound in the classical pathway. Well, by the time we get C3 present, we can now turn on the alternative pathway too and get tons and tons of C3 on the surface of that cell, so that we can do things like get holes all over the place on the surface of that cell. So not only is the alternative pathway really great because it is super, super, super fast. It's basically instantaneous when those microbes come in. It also can serve to amplify the other aspects of the complement pathway. There is a problem with the alternative pathway. I'm going to tell you the problem with the alternative pathway, and I'm going to leave you hanging over the weekend and give you the solution on Wednesday, or yeah, you could look at the textbook. But the problem with the alternative pathway is I showed you this sort of automatic binding to the pathogen surface with the RNH2 or ROH group. The thing is, all of your cells also are covered with these things. And so the alternative pathway, not only is the alternative pathway binding to any microbial cell in your body right now, it's binding to lots of your cells too. Your cells are also getting C3b stuck on them right now. And so how in the world we destroy just the bacteria without destroying all of our cells is the challenge of the alternative pathway, and the thing that we're going to start with on Wednesday. So I promise that you will not be lysed by your membrane attack complexes between now and Wednesday, and I will see you then. Remember that there is the Inquisitive by 5 on Wednesday. It's all questions having to do with aspects of complement and antimicrobial peptides. So have a great weekend.